Hello, my name is Mitch DeVolder, and I'm going to present my current project modification of Dnase1L3 and Aerolysin by Quickchange Mutagenesis. So necrotizing fasciitis is a serious infection of human soft tissue that is caused by toxins released from various bacteria. Some such toxins include aerolysin, streptolysin, and perfringolysin. These toxins are the key virulence factors responsible for these infections. Today, I'm going to talk mostly about aerolysin. So Aeromonas hydrophilia is the bacteria responsible for production of aerolysin. Aeromonas hydrophilia is a gram-negative bacteria that works in coordination with other bacteria to put together an attack against host tissue. Aerolysin is the PFT, or performing toxin, produced by Aeromonas hydrophilia that causes cell lysis. It does this by inserting a pore in the host cell plasma membrane that allows for the cellular contents to leak outside of the cell and eventually causes the cell to die. Studying how these toxins bind to the plasma membrane of host cells is not well understood and is difficult, but Quickchange mutagenesis allows us to more accurately study these interactions. So Quickchange mutagenesis is a biochemical process that allows us to introduce mutations in the form of substitutions, deletions, or insertions. These mutations are then amplified by PCR and then purified using a DPN digest and then plated on a petri dish that is treated with antibiotics that will select for only the cells that take up the mutated plasmid. The first step in Quickchange mutagenesis is designing the primers. These primers have to be designed with the melting temperature in mind to allow them to cooperate for PCR. So the melting temperature, as you see here, is close to 78 degrees, and this allows for it to bind and unbind to DNA as desired during the PCR process. When designing this primer, you pull a section of DNA from the gene of interest and pick out the exact nucleotide sequence that encodes for the amino acid for which you hope to change. This is an example from aerolysin, where we are mutating a lysine to a cysteine. So the AAG becomes a TGC. As you can see here highlighted in yellow, this is the new TGC mutation. These primers come in pairs because when the DNA separates, you have a forward primer and a reverse complement primer. And these primers are important in the PCR process. During the PCR process, just like any other PCR process, you have an original DNA sample, the primers, the DNA primers that contain the mutation, free nucleotides, and a DNA polymerase enzyme that's not pictured here. During the first phase of denaturation, the solution is brought to a temperature of approximately 95 degrees that allows for denaturation, and the double-stranded DNA becomes single-stranded. In the next phase, the temperature is lowered to about 55 degrees, and this allows for the primers to anneal to the single-stranded DNA. And then the temperature is again changed and risen to about 68 degrees, and this allows for DNA polymerase to become active and elongate the rest of the sample. This process is repeated about 20 times to allow for amplification of the new mutated DNA product. As a result of PCR, we will have a piece of DNA that contains the mutation and some DNA that does not. The pieces of DNA that do not contain mutations will be able to be digested by DPN, and the resulting solution will contain the mutated plasmid as well as some other fragments of DNA, and these DNA fragments will be washed away using a PCR cleanup process. Once we have the purified mutated plasmid, that plasmid can be joined with some competent bacterial cells in a tube and exposed to high levels of heat for a short period of time, which will cause a stress response of the bacterial cells that will elicit them to take up the plasmid from the environment. After doing so, those bacteria will be plated in a petri dish that contains an antibiotic, and this antibiotic will correlate with the antibiotic resistance gene contained in the plasmid. This will allow for only the bacteria that took up this plasmid from the environment during the heat shock phase to grow up on the petri dish. After the cells grow up, they will be lysed and their DNA extracted using a mini prep. During this process, like I said, the cells are lysed, and then that solution is added to the spin column you see here, and a DNA binding solution is added that binds to the DNA, but allows for everything else to be washed through during centrifugation. That solution is washed a few times, keeping the DNA in the spin column. And then finally, an elution buffer is added to the spin column, which will allow for DNA to continue through the spin column and into a clean tube. So finally, we have a solution that contains just the mutated DNA, and that solution can be sent for sequencing. This process you see here is Sanger sequencing, and the results will be able to give us an idea of whether or not the Quickchange process was successful. And we are able not just to make sure that the mutation of interest is present, but that others are not. And reading this, you can see that there are peaks for each respective nucleotide that occur at greater strength whenever that nucleotide is present. So in conclusion, I was able to successfully create several mutations in aerolysin, streptolysin, and perfringolysin using Quickchange mutagenesis. And these mutations have contributed to the testing and understanding of various membrane molecules' importance in the binding of pore-forming toxins. These mutations, also created in Dnase1L3, have been used to test for effect on catalytic activity. Lastly, I'd like to acknowledge my PI, Dr. Peter Kyle, Minal Engeval, Brittany Mapp, John McCord, Brian Sutton, the rest of the lab members in Dr. Kyle's lab, Texas Tech ASM, and True. Thanks.